Okay. Can you guys hear me? Okay, cool. Thank you, Teal. Confirmation that I'm not just talking to no one. Great. Okay, I will get started then. So today, hang on one second. Yeah, I can't hear anyone else. But that's okay. You can type, if you can add in questions or type into the group chat box, then I can see those. So Teal, what you just put in as questions, I can see. And I'll answer those as you go. So feel free to interrupt via text or voice if that's possible in this format. So today I'm going to talk about PhotosynQ, which is a platform to enable anyone to quickly and easily create, share, and analyze plant health information globally. And I want to talk about sort of some of our goals, what the concept is, what you can measure, some of our goals, and then I really just want to dig into applications for ag extension agents and even maybe create a quick project. So as I'm going through this, if folks out there could think about a project that you might actually want to run, because in about 10 minutes, I'm going to create a project. And it would be more fun to create a project based on what you guys are actually interested in, just for fun, rather than me making something up, which I will do if I have to. So let's get started. PhotosynQ is a project that's run out of the University of Michigan. We have a couple different funding sources you can see there. And like I said, our goal is to make a big global database of plant health and help people answer big global questions about plants, sort of create the platform to enable those types of questions to be asked, whereas now they can't be asked. So what are we trying to do? What's our goals? Well, one of the things that we'd like to see, some of the outcomes, is we'd like to see a 10 or 15-year-old kid ask an interesting question on the Photosync platform and have people all around the world help him or her answer that question. So that's sort of democratization of science. We don't just want professors to be able to ask interesting questions, but we want everyone to be able to ask those questions. And we want the quality of the question to dictate how many people get involved and to what extent. So this is a non-hierarchical platform. We want to make a low-cost device that people, even in the developing world or even here in the US that don't have access to a lot of money can use. So this is Kelvin. He's actually here at MSU, but he's from Zambia. And he does a lot of breeding on the common bean. And so he's going to use this device in Zambia to hopefully improve his breeding efforts and do marker-assisted selection to speed up the time in finding a drought-resistant bean for him. So that's another sort of goal, is to get more of these devices out in the world to people who otherwise couldn't afford it. Another goal is to get more people involved in science. We would like to publish a paper with 500 contributors, where most of those folks were people out collecting measurements as part of the project. And we think that will happen, actually. And then finally, we want to, sort of the biggest, broadest goal is to answer big global questions about plants and plant science that we can't currently answer. So I'll give you an example. Right now, there's ways of estimating photosynthesis rates from satellites. But this measurement's never really been confirmed with actual ground measurements. So, one big question we could try to ask is to ground truth these global photosynthesis measurements from satellites to see if they're actually accurate or not. That's just one example, but you could imagine lots and lots of examples where if you had thousands of people with high-quality scientific devices all around the world, you could start to ask and answer really interesting questions. So we have this is old actually, we have about 150 members on the site who've taken measurements. We've taken about 20,000 measurements so far, most of them this summer. And I'll show you some of those projects as we go through. And I know for some of you who aren't familiar with the platform, I haven't explained it really well yet, but we're actually just going to walk you through the process. So don't worry too much about that. Here's the device. It's all 3D modeled. It's not the prettiest thing ever. It is a beta device, but it works quite well. We've gone through a lot of iterations, and we've done a lot of testing on it. It has a bunch of components, and I just want to sort of go through them quickly. It has temperature and relative humidity sensors. Those are ambient temperature and relative humidity, not like temperature of the leaf at this point. CO2 sensor, which is also an ambient CO2 sensor. And then a light intensity sensor, which is PAR. PAR is photosynthetically active radiation. That means the portion of the electromagnetic spectrum which is used in photosynthesis, or which can be absorbed by plants to do photosynthesis. That's about 400 nanometers to 700 nanometers. So it's a little bit more specific than your standard off-the-shelf light sensor. So we can measure those things. We can also measure specific photosynthetic parameters. Some of you probably have some experience with this. Minolta spad meters, very, very common for measuring chlorophyll content. So we can also measure the chlorophyll content. Phi2 or FV over FM, that's photosynthetic efficiency of photosystem 2. We can measure that. We can also measure some really fancy things like proton motive force, which relates to ATP production. It's the closest thing that can measure that is a desktop unit that costs about $50,000. So we can measure things. If you took all of the things that we can measure in this handheld device and added up the cost of the equipment you'd need to buy, it would be in the tens of thousands of dollar range. Just Minolta spad meter itself is $2,500. So you can see on the left, if you're sort of familiar with spectroscopy, we have 6 specific separate LEDs at different wavelengths, and then we have 2 separate detectors which pick up visible light only and then an infrared light only. That's how we're able to take these measurements over here. So what do these things mean? What do they relate to? Photosynthetic efficiency relates to stress, specifically drought stress, but also nutrient stress. Chlorophyll content can also relate to stress. I mean, it's basically a leaf greenness, but it can indicate when you should add more fertilizer. It's used for corn for that purpose. Proton motive force is a little less clear at this point. It's a very specific measurement in terms of the mechanism, but basically the background work hasn't been done on it yet to understand how we can then apply it to an actual situation to make a recommendation to a farmer or something like that. But we have very high hopes for that one. And then finally, using the CO2 sensor and these tools, we can measure some different measurements from the soil. So we can measure soil biological activity. And you can see this middle example where we have a device sitting inside a little container. What you do is you put the device on the dirt and you seal it over top of the dirt, and then you measure the CO2 as it builds up in that sealed container. And the rate at which the CO2 builds up tells you something about the biological activity in the soil. We can also measure soil active carbon or humus levels using a standard method which uses potassium permanganate. That does require a little bit of pre-work. It's not just a spectroscopic method. You have to have a cuvette and mix some reagents, but it's pretty simple. We've done a couple of times in the field. And then you can attach other probes to this. If they're outputting a simple analog signal, you can attach things like soil EC probes or soil moisture probes, things like that. And we've done that. This is an example of a very simple soil EC probe that we attached. Okay, so that's the capabilities, and please throw questions at me if you have questions about the capabilities, or wait until the end. Either way is fine. So we've done this all for under $100. Now that's parts, right? If we were a company, it would be parts times 10. Our goal is not to charge a lot for the equipment, but our goal is actually to get a lot of devices into the world. So we're trying to keep our price point around $100 or $200. Okay, so who is this useful for? Well, we are a plant science lab. I work in Dave Kramer's lab. He's developed a lot of the conceptual framework behind many of these photosynthesis measurements. So we're a pretty hardcore plant science lab. For us, it's useful because it's less expensive, so we can have more devices, take more measurements in the lab. The quality is really good. This was built sort of designed off of a benchtop device, which Dave's been working on for 20 years, which is $10,000. The quality on this is pretty close to the quality on that. It's improvised trainings. Grad students come in, they can take them months to get familiar with how to create a new measurement or build protocols. We have tools in the PhotosynQ platform to make that really simple. And then finally, it is modifiable. This whole thing is open source. You can take the hardware, modify it, you can contribute to the software, you can develop new methods, you can slap on different tools. It's built for that. Educators, so this last fall, we actually ran a project, or we didn't, they ran it. A teacher who was teaching an intro biology class here at MSU ran a project where the students collected a couple thousand measurements over the course of the fall. You can see some of the data here. This is actually from the website, different data points. For them, it's great because it's low cost. You know, the kids were using it within 30 minutes, collecting quality data. And it's project-based learning that connects to real science. Our first paper that we're going to publish will probably have those kids as co-authors because they collected data, or at least reference. We do also have a community. We're actually going to get the devices out in the world through a group called publiclab.org. And they're sort of environmental activists with a focus on promoting sensor-based journalism. So they're interested in this device as well. So, and then finally, plant breeders. So in the example I gave before, Kelvin in Africa, he's a perfect example. So for them, they want to take thousands of measurements in very short periods of time, which is something that we can do. They have technicians out taking the measurements who can change, so it needs to be easy to use, which it is. And then they can literally view and analyze their data the same day they collected it, which is just kind of unheard of when most people are still writing things on pieces of paper, transferring them into Excel files, and then sharing them via email. So those folks are pretty excited about it as well. So now let's get to what extension can do with it. I think that's been enough intro. So I'm going to create a project, and then we're going to collect a measurement as part of that project. Then we're going to go look at the measurement in our Data Explorer, sort of data analysis tool, online data analysis tool. We're going to talk about the sort of quality of data that we can collect. I want to show you some examples of some pretty that I think will be interesting to you. And then I'll talk to you about some of the features that we want to add in the future to the platform that are especially relevant to extension. So let's go create a project. Let me stick this over here. So first off, does anyone have a project that they would like to create? It could be something like, maybe you're an extension agent in New York and you want to track the health of apple trees, in one county, in the county that you work in, or you want to identify if one management practice is different than another, or something like that. Do you have any suggestions? If you do, put them in the questions box. However Teal did that, do that. I'll give you a couple seconds there to figure that out. Okay, I'm just going to make one. I'll make it up as I go. So this is the webpage, photosync.org. Here's some existing projects. Again, we've already taken a lot of data here, but we're actually going to create a new project. So I'm already signed in. I'm going to go to create, and I'm just going to fill out my project here. So I'm going to do the one I just said. I'm going to track. I'm going to track the effect of management on apple trees. Right, and then here in project description, I would tell people about the project, in New York. Here, explain management practices, et cetera, et cetera, et cetera. You'd upload a photo, and then you'd give people directions. Now, different people use the platform for different things. Some people are creating projects in which they're the only people contributing. It's just their own little project in their greenhouse. That may be you, or you might want to create a project where you've got a bunch of people contributing from all over the world or from all over a county. So let's imagine in our apple tree case, we have 7 different extension agents who are going to be collecting data using a device. And so it's important if you're working with other people to give really, really clear directions, because the quality of the data that you get back is completely dependent on the sort of training and ability to follow directions of the people who are taking the measurements for you. So we're going to give good directions here. I'm not going to write them in, but that's really important. Then you can say, can other people contribute to the project, yes or no? In our case, we want that because we've got these magical 7 other extension agents who are going to be collecting data. Now, this is questions for collaborators. Imagine this is like a survey that every time someone collects a data point as part of your project, they're going to have to answer these questions. And these questions are absolutely critical. You need to really think deeply about what they're going to be, because your ability to analyze the data at the end is completely dependent on how you tagged the data or the types of questions you asked. So in our case, we're going to have species of apple tree, right? Or not species, but well, whatever. So so we're going to have like Gala and Jonah Gold and Macintosh. Okay. And we're going to have treatment type. We're going to have, maybe this is, we had four different management practices. That's what we should call it, management practices. So we have, 4 different management practices. And then we're going to say, who's the farmer? And then maybe we have 5 farmers, Bob, Karen, Joe, whatever, it doesn't matter. And then maybe I'll leave one more that's, I'll just say note. And note I'm going to select as user defined. So the rest of these, I've defined the answer choices for the user. It's either going to be Bob, Karen, or Joe for farmer. But in the case of this one, the person collecting the data is actually going to have to write in some value, which is not predefined by me. This is useful sometimes. If you don't know all the answers, you want people to be able to write things in, you use this. Okay, so we have our questions defined. Again, this is important to think through thoroughly before starting. Now we're going to select what measurements we're going to take. And let's do fairly simple ones. We'll do our chlorophyll fluorescence, that's photosynthetic efficiency, and then SPAD. We will have more videos about sort of what these are, which ones you might want to use in what situations, all that kind of stuff. And I'm not going to go into detail here, but yeah, we'll pick these 2. And then I'll sort of show you the results. Okay, and then this disclaimer basically just says that everyone's going to be able to see your data, which is the default right now. That's the only option you've got. So sure, everyone can see my data. So now I've created my project. Cool. Other people can join the team and contribute to the data. So now let's, so is there any questions so far? If anybody has questions, throw them up on this list. Of questions. Okay, I don't see any. Let me make sure there's none on the other forum here. So, okay, good. Okay, so now we're going to take a measurement. So, let's see here. I'm going to show you. That's me. So I'm going to show you the device. So this is the actual device. It's actually 3D printed. There's only going to be a few hundred of these beta devices. The next version will be professionally manufactured. And the way that it works is you take a plant like this one. Set it here. So you take a plant like this one, and we're going to clamp the leaf. Press go, and that's going to take a measurement. We're going to get our light intensity from this sensor right here. It's going to sort of match the light that is naturally seen by the leaf, and that's the result that we're going to see. So let's go do that. You have 2 options. You have 2 options for collecting data. You can collect your data through, one second, I'll pull this up, through your phone. We have an Android app for that. So here's sort of the phone interface. And this is what you would use out in the field. The phone has the ability to cache data, so you don't really have to worry if you're out of Wi-Fi range, no problem. The data will get cached. When you get back in range, it'll automatically upload the data. So I just wanted to show you that we do have an Android app. It is only Android, no iPhone, but we will eventually develop an iPhone app as well. But I'm actually going to collect the data on the Chrome app. So this does the same function. It connects to the device I just showed you using USB. Using USB. Hang on, I need a USB cable here. Can I have a USB cable? Oh, you're in the middle of a, oh, shoot. That'll work, perfect. Thank you, sir. Sorry about that. Wasn't prepared. Okay. So you connect to the device using USB, and so you can sort of see what's going on at the same time. So right, I'm connected via USB, and I connect to the device here. It's very simple. Multi-spec ready. So it says, yep, I'm ready to go. We can go over here to projects, and we can go through all the projects on the site. And the project I just created, I can find it. Just search for Apple. Oops, I'm sorry. Got to refresh everything. Let's try that again. So there's my Track the Effective Management on Apple Trees project. I select that. I can see all the questions that I asked here. Species, management practices, farmer, etc. So I'm going to take an example measurement. So here we go. We'll take a couple even. First. Okay, so we're all connected. I've answered my questions. If this were a big project, I may have had to follow specific directions. You may have had to told me, climb up to the top of the tree, do this, do that. These questions may relate to other things, like they may relate to pests, count the number of pests on a plant. They may relate to seed pod counts. Whatever you want to put in there is fine. Okay, so I plant my leaf. I press start. It's showing that there was 2 protocols, and now it took the 2 protocols. So here's our data. This is our chlorophyll fluorescence. This is our chlorophyll content. The key data points here are Phi2 and SPAD. Phi2 is the portion of incoming light energy that's going on to do photochemistry, as compared to the portion which is rejected by the plant, fluoresced back out. This is just an arbitrary number, basically, an arbitrary value defining the greenness of the leaf. So this leaf's pretty green. The Phi2 is not that high, given that it's indoors. And you'll learn basically what these things mean over time as you take more measurements, and you'll sort of understand this stuff. So once we've taken the measurement, we click on Keep. That submits it to the website. You can see we successfully contributed the data. And now we can go back to our project page, and we can see our measurement. So this is the data that we just collected. But much more interestingly, I can click on Dashboard, and then I can see my data in lots and lots of ways. I can map my data. This one's, because I took it on a computer, it's not going to map. If I had taken it with my Android app, you would see a map, and I'll show you some examples of that. I can download my data just as a CSV file or as in an Excel file, and I can plot my data. So I just wanted to show you we can very, very easily go from creating a project to collecting data. That was 5 minutes. Now I want to go show you some actual projects that have interesting data. So let's go. This first one that I'm going to show you is, here's one. This is one that Dan, who works in Malawi, collected a week or 2 ago. So he was in Malawi, and they were interested in nitrogen management in conservation ag systems in Malawi. So let's go look at the map just to see where we're at. Now these ones were taken in the field using the Android app, and so we do have GPS data from on satellite, and you can see the field that this was made in. How cool is that? Not only can you see the field, but we can actually go through these were all user questions, the same way that we had our apple species user question. He had user questions about the crop type and district. I'm sorry. Yeah, I'm done with that. Crop type and district, fertilizer rate, things like that. So let's see. We can select different series. Like we can say, I just want to look at block 1. So now I can just see, I can look at just block 1, which is kind of buried under there. But let's do this. Let's look at every possible combination of all of these different questions. So that's every possible subgroup. Now we're starting to see something interesting. You can see that the field was partitioned based on these subgroups from top to bottom. So that's the mapping feature. You can go to plot, and you can plot things like, how does light intensity relate to photosynthetic efficiency? A little hard to see here because let me delete all these things here. So let's do that again. How does light intensity relate to photosynthetic efficiency? Zoom in. Well, you can't see it very well, but in fact, as light intensity goes up, photosynthetic efficiency goes down. And you can see it a lot more clearly if you do linear electron flow by light intensity. You get this very, very nice curve. I know this is a little bit technical, but it's actually quite important. So this curve is telling you for these plants how healthy they are. And when you see points that are way off this curve, either there's something wrong, and we can check for that. So here's an example point, like what's going on with that piece of data. It's certainly not in line with the rest. We can click on it. Oh, shoot. It's been doing that on me. Anyway, we can click on it and see the specifics. But I just wanted to show you this as an example. So let's go look at some simple stuff, like how about averages? Let's look at the different crops. We have 2 crops. We can look at the average linear electron flow for each crop. We can look at the average SPAD for each crop. Now the trick with this stuff is that there's actually a lot of variation, right? And you'll find this. In these fields, there's a huge amount of variation. So to try to pull out anything specific is quite difficult. And so for you guys, you're going to want to use a little bit of statistics to be able to say something interesting from what is otherwise fairly messy data. So I want to show you an example project that we've done that on. So I'm using a project, a program called StatWing. It's very easy to use. You can literally just download your data like this, click Save, save it as a spreadsheet, and push it directly into StatWing. No fooling around needed. And do some pretty neat analyses. So this was the data from a guy who was looking at boron, which is a micronutrient, and its effect on stress, drought tolerance. So he had 7 different treatments, different boron treatments, and he wanted to see if that would affect chlorophyll content and photosynthetic efficiency. So we took his data and we put it into what's called a regression model, where we try to predict the chlorophyll content, that's the Minolta SPAD here, based on the factors that we think will impact it. So you can see here that Minolta SPAD is higher in the middle and bottom leaves, but is lower in the top leaf, which makes sense. The top leaf has the most sun. It tends to be the most, the lightest. You can see that there's a slight effect, but not really significant, of light intensity on SPAD. So that means that leaf age has a bigger effect on chlorophyll content than light intensity, which is interesting. And then here's all of his different treatments, T1 through T7. His predicted best treatment was T6. That was a combination of boron plus sorbitol that he thought would absorb better. And you can see that, sure enough, T6 shows a significant improvement in chlorophyll content as compared to all the other treatments. Those are not significant. So we were able to get this data within 5 minutes of him taking his last measurement. It was very, very, very simple. I'll show you another example. So this is photosynthetic efficiency. Again, it relates strongly to light intensity. It also relates to leaf age, if it's the top, middle, or bottom leaf. And again, his expected winter treatment shows a significant difference, meaning it has higher photosynthetic efficiency. If we were to look at the same project just in terms of averages, average values between T1 and T6, or T7, you would see no difference at all. And if you were to look in the field, or in the growth chamber in this case, you would see no difference at all. These are not visually noticeable differences. You have to run statistics to see it, but Photosync gives you the capability to, the purchasing power to buy enough devices, the ability to collaborate easily, and the ability to collaborate over large distances so you can get the kind of data you need to see small differences between treatments or between populations. So that's the thing that I think really has a big positive impact for the case of extension agents. So that's kind of what I wanted to show you guys. I think I've gone on long enough. The last thing I wanted to say was, I expect that people will ask, well, can you give sort of a direct sort of policy, no, policy is the wrong word. Can you tell somebody what to do directly based on the data, right? So they go out and they take a measurement and it says, hey, there's too little water, or there's too much, fertilizer, there's too little of this. And the answer is right now, no, but that's something that we are working on. So in order to be able to do that, we need enough data so that when they say, I am growing corn, it's week 6, there's been no rain in the last week, and it's this variety and this location, right? If they can say those things and we have sufficient data to try to match existing known populations with known outcomes with their specific case, then we can build an algorithm to tell them, hey, given all these other guys who are similar to you, you have very low chlorophyll content, or you have very high chlorophyll content, or this looks healthy or not healthy. Then we can give immediate feedback to farmers. That's kind of like the long-term goal. But it requires a lot of data to be collected first. So anyway, I just wanted to preempt that question. So that's it. There's a lot of potential applications, and I guess I'll just sort of open it up to questions if I miss something, or if you have specific questions. Go ahead. I think you can ask them in the Q&A. Is that right? Is there anybody that has questions or comments or thoughts or applications? I would love to hear about applications. So someone asked, can you make the projects open? Yes, you can make the projects open. Anyone can contribute. That's the default state, actually. Yep. Okay, cool. Well, if there's no other specific questions, feel free to shoot me an email if you have any ideas, or we are in beta right now and we have a lot of devices we need to ship, but if you have a really interesting application, let me know and we could try to add you to this year's beta. And we'd love to do that. We have a lot of projects going on relating to the common bean, to wheat. We're hoping to do some stuff with grapes. So if any of those things fit you in particular, we already are working on projects for that, so that would be a good fit. But yeah, otherwise shoot me an email or go check out the site, and feel free to browse the data and see what you see. So unless there's anything else, I think I'm gonna head out, and thanks for coming. Oh, I should put up my email. I don't know if you guys can see this. Okay, thanks again. Oh, wait, I got one. You try tracking the effects of diseases through a season. No. So we've only had 1 year's worth of work on this, and that was a year in which we were also working on the hardware and the back end and a lot of other stuff. So I can show you a little bit of that data, but the answer is simply no. So that would be a really interesting question. And in fact, that was somebody brought up an extension agent here at MSU was talking about aphids, I think. She's talking about aphids, and she was saying it wasn't clear why aphids attacked at certain times or certain places and not others. And some people thought it was sort of high nitrogen in the leaves, and some people thought it was high sugar content, and it wasn't clear. So an interesting application here is if you had a county or a state where extension agents had these devices in their hand while they're just walking around, and a farmer says, I think that this gets, so the next question. In terms of the post on YouTube, I think that this gets saved anyway. So I think you can come back to this page and it'll just play it automatically, but I will check on that. But to go back to the disease thing, imagine, farmer X says, crap, I've got a bunch of aphids. So extension agents are out there anyway, and they start to take specific measurements as part of a project to track aphids, right? But in addition to that, they're able to measure nitrogen content in the leaf, and in fact, we could measure something like Brix or sugar content in the leaf if we modified the device, which is possible. So you could as soon as you saw the effect, you could start to measure it in real time all over the county. You could see the movement of aphids, and you could see if aphids were moving along lines which were consistent with high amounts of nitrogen or sugar, right? So that would be a really cool project, but you kind of have to have the devices in the field ready to go to do it. So I think tracking diseases is a really, really good idea. Thanks, Jim. Yep. Cool. Any other questions or thoughts? Okay, cool. I'm going to head out. Thanks, you guys, for coming. Send me an email if you have anything interesting. We're always here and love to hear from you. Bye-bye.